I'm here as the previous editions, and I'm delighted to show you this data here. And this is just a break from the embryo, because after me, I think that Christina is going to come back with the embryo. So just you are going to have all of this from the maternal endometrium, so I just want to keep your attention, because this is going to be the only part for that. First, I would like to disclose that I am the scientific director of Igenomics, which is a company that aims to translate basic research to the patient through clinical trials after proving that it works. So this is, and some of the things that I will discuss, they will be related. And I would like to start with the endometrial factor, which is what I'm going to tell you about. And I'm going to start just with the last word of the previous presentation, which is exactly that. This was the question from Christina, the last question. Unlike many mammalian species and even non-mammalian species, we humans, we have this higher aneuploidy rate. So this is something that accounts for the problems with the embryo and why human reproduction is not as efficient as we would like to. And that's why we have been focused on the embryo that much for the last 40 years. But even so, even if we consider euploid embryos, I mean, taking as a natural conception, we know that only 35% of implantation rate exists. But even if we transfer euploid embryos, we know that this is not 100%. In fact, this will be about 60% in randomized clinical trials. More than that, could be different centers, different studies, whatever. But so it means that there is still a long way to go. And if we compare that with rodents or rabbits, you can see that they can achieve easily 95 and 98, even if they do not have any chromosomal aneuploidy. So why is that? So the main reason for that is because human implantation is different. Unlike the other species, we humans, you women that menstruate, you are on control of the implantation process. It's the maternal part that controls the implantation process, unlike rodents, that is the embryo that is really controlling that. In fact, the adipose, we know for a long time in many species that the embryo can float around for a long time until you give a shot of estradiol in a mouse or in a marsupial. Now they are ready to go ahead, and then the embryo implant and goes. This doesn't happen in humans. So the whole thing to have menstruation is to prepare the endometrium and to prepare the window of implantation. And if it doesn't work, you restart the cycle again. So this is for you to keep in mind. And in fact, I call this as the first step of the journey of life. Of course, for many of the audience will think that the first step is the embryo. But I will point out that the first step is a receptive endometrium, because the embryo is able to implant and to adhere wherever it wants, including a plastic pipette, a plastic transfer catheter, but not in a non-receptive endometrium. So if there is a non-receptive endometrium, the journey will not start. And this is what I would like to try to convince you in the next 30 minutes. First, this is the concept that there is a specific moment for implantation to occur in humans. And this is called, what you know, is the window of implantation. This concept came from a long time from this work of Hertig and colleagues, that they were able to flash those embryos at different times after they have the intercourse, and they were able to see that at a specific time they were not able to obtain the embryos they already attached. So this was just the very first observation. But it was David Nabot in the 90s that, using the ovum donation program, demonstrate without any doubt that when they transfer, at that moment they were transferring embryos from 2 to 12 cells. And when they did it from within days 17 to 19, they have what was the normal implantation rate at that time. But there was not a single pregnancy when the embryo was transferred before, on day 16 or day 20 onwards. consider that this is, again, the window, the clinical window. This is the moment that where the embryo were transferred. Of course, the embryo must develop to the blastocyst in order to implant. So this was the very first proof of concept for that. But it was Wilcox in 1999 that popularized this concept. This was a nice paper in New England Journal of Medicine, and they popularized the concept that the human embryo implants from day 8 to day 10 after ovulation. They achieved this conclusion by following ovulation with an assay that never was used in clinic, which was to measure those metabolites of estradiol and progesterone. But by doing this, they get to the point that within this time frame, embryo implantation or the window of implantation was present. But this was the observation. Now, what comes after that is that we believe that there is a wide time frame that always the mother will be for us, regardless of we have a natural cycle, control-stimulated cycle, or an HRT cycle, and regardless whether there was individual variation. And this is why I think that the endometrial part has been somehow neglected in the sense that say, okay, let's focus on the embryo because the window is there and we will transfer the embryo and we will give progesterone. That's it. That's what we used to do in order to say it will work, okay? So let me give you the whole view of what I think we should consider also because we are not achieving any more than 60–70% just focusing on one factor. We have to consider both. So for the endometrial factor, we have had the histology, which was the pioneer work from Noyes in the first issue of Fertility and Sterility in the 50s, histological findings, and we can just know when, the dating of the endometrium. This was fabulous, but it is 70 years ago. And since then, nothing came that much in order to understand endometrial physiology. We have immunohistochemistry. We have single molecules that try to develop in the 90s, but I think that it didn't prove successful at the clinical level. Ultrasound we are using, and this is our way nowadays, still the only way really to assess endometrial receptivity, among brackets. Doppler is there. And hysteroscopy, you know that we are using this whenever, when everything is lost, when we have an implantation failure, when we don't know what is going on, we do the hysteroscopy. So this is what has been done so far. But nowadays, we are at the moment of the genetic revolution. We are at the moment of artificial intelligence, machine learning. We have iPhones, all of us, many millions of circuits. We know that we cannot rely on single molecule or single circuit. So we have to go with our times, which is basically the genetics, and in this case, the transcriptomics, the secretomics, stem cells, they are getting a place in the understanding of the development of the endometrium. And recently, the microbes, the existence of a microbiome in the human endometrium is getting also some presence, and it could be somehow diagnostic application. So all what we have done until, all of this must be considered together, but I think that we have already tried for a long time this part, and I think that we have to try also, and we have to know more about what could be used to improve our efficiency in the diagnosis. So I would like just to mention about that. I think that the most reliable technique, an extended technique that we are using nowadays, which is the ultrasound, that basically is our eyes, we are aware that the fact that we have a triple line endometrium 7 millimeter doesn't mean that it's receptive. It's nice to have, but there are plenty of data that shows that really this is not the way that we can predict implantation. Because we have to jump from the anatomical medicine to the molecular medicine, as in other fields of medicine, they have been doing that for a long time. So that's the thing that I want to mention about that. So again, we are in the area of genetic revolution. And in fact, I think the last lecture today is about that. Transcriptomics is the obvious, is the most advanced technique. You know that in oncology, every cancer comes with a transcriptomic signature, and it makes the difference between one and other treatment. Histology is nice, but this will just add the value that it needed. So this was the idea, to add these glasses, these transcriptomic glasses, to the human endometrium. And this is what we and other groups have been done for the last 10 years, which is instead of trying to develop just the histology that has been for a long time known, let's go to objective criteria, let's go to transcriptomic signature that can let us know whether this is receptive or not, and when it's going to be receptive, and can be interpreted in an objective way using also machine learning, and then learn more and be more accurate on that. And this is the concept of all of this research. We launched this endometrial transcriptomic signature in '91, and we made a test of that. It calls the endometrial receptivity analysis nowadays, and it's made of these 236 genes, the transcription, the expression of those genes. But the important thing are not even the genes, are the relation between them, and there is a bioinformatic predictor and a classificator algorithm, and at the end of the day, with this system, allow us, when we receive a sample, without looking for the histology, we know whether if this endometrium obtained at this time from this specific patient is receptive, or the window is already gone and we have to obtain before, or it's pre-receptive and we have to wait, we have to give more days of progesterone in order to have the window at this time. And of course, the system has been improved, and the system allows us, within the window of receptivity, to go in the center and be even more accurate, going to the optimal part of this. Within this concept, what we have learned in the last 5 years that we are doing this, or 6 years, is that there is no such a thing as a window for everybody. The majority of patients, 60% of them, the window will be after 5 days of progesterone. But you have between 30 and 40% of patients that the window will be a little bit delayed, and some of them, they will be advanced. And some of them, they will have even a narrow window of implantation. All of this is personalized information that you can add to the treatment, improving those cases, specifically the most difficult ones. And this is what we have trying to work and demonstrate about that. And by learning this, the idea will be, it's to develop the concept of personalized embryo transfer, by which you can transfer your blastocyst in this case, not at the moment that your embryo is ready, but at the moment also that the endometrium of your patient will be receptive. And this is what we call about personalized embryo transfer. Data has been accumulated, more than 20 papers, not only by our group, by other groups worldwide, trying to prove whether this system could be diagnostic or not. And so far, we have analyzed more than 32,000 endometrial samples worldwide, more than 60 countries, and the numbers are this. So about 70% of cases, 66% of cases, they were receptive at the moment that the clinician thinks that should be receptive or should transfer the embryo. But you have this percentage, this 29%, 30% that they are not, and this is what makes a difference on that. We start from case reports of the most difficult patients, 7 previous failures, and then finding out that the window was displaced, transferring the embryos, and having the results later on, through prospective clinical trials, comparing patients with recurrent implantation failure versus controls, through basically randomized clinical trial that we have just finished 1 year ago. And in this clinical trial, it was set aside about 546 patients. These were the inclusion criteria. In these patients, PGS was not an inclusion, neither an exclusion criteria, so we tried to just assess the endometrial factor, but we don't know about the embryonic factor. So, but we hope that in a RCT, you correct the other factors, because you just select for the endometrial factor. This is the design. This was the design of this study. We have blastocyst transfer either with fresh embryo transfer. They are assigned either to a fresh embryo transfer directly, or we freeze them all and we do, after that, a deferred embryo transfer, or they went to the arm of personalized embryo transfer, by which we freeze them all, we do the ERA test, and then we transfer the embryo as indicated by the ERA test. This study was started in 2013. The interim outcome was shown in 2016, not showing the surrogate markers. First objective was live birth, but we were able to display in the interim study implantation rate, pregnancy rate. Obviously, we cannot display a live birth because this was in the mid of the study. So the data that I'm going to show you was presented in ISRM, and it was about these 356 patients, more than 100 patients in each arm of the study. And the study, and this was the centers participating worldwide in that, and these were the results, again, from the interim study of the RCT, in which what was statistically significant, this is the pregnancy rate, fresh 61, all frozen 60, and personalized embryo transfer 85.7. Implantation rate was increased, but in the personalized embryo transfer group, but was not significant. The losses were similar. The ongoing pregnancy rate also increases in the personalized embryo transfer, but was not significant at that time. And interestingly, we have less twins and more singletons in the personalized embryo transfer group, but was not significant. So these were the data of the interim. The study finished 1 year ago. We are just collecting the data from the live birth center by center, and hope that this year we will be able to publish the final results concerning this. Again, this is for the first appointment. This is in the case that the patient comes. Nowadays, it's being used for patients with implantation failure, the most difficult patients. This randomized clinical trial was performed to assess whether it will be cost-effectiveness to do it in the first appointment, as we do the semen analysis or the AMH, and we will see what comes after that. This is a study from a group in Canada. This is independent for us, and they want just to check what could be the contribution of the endometrial factor on top of the embryonic factor. They were just transfers of euploid embryos, either in a standard embryo transfer or in a personalized embryo transfer after doing the ERA test. And this is what they got in terms of implantation, ongoing, and live birth rate. So there was a 20% increase in implantation rate, similar in ongoing pregnancy, and this in the live birth. They were not statistically significant because this is a retrospective study with 78 patients, but I have to show you this since it came, and also can show you nicely what could be the addition of the endometrial factor on top of the embryonic factor. But again, these are retrospective data. These are our own, again, retrospective data from display from 11 clinics worldwide, and this is business as usual for them, and this is the result of doing this is after they transfer in the first attempt when the ERA was receptive or when they corrected the window of implantation. And this is the number of personalized embryo transfer performed, 500. This is the implantation rate, which goes about 56, 58%. This is the pregnancy rate, 73, 72%, and these are the rest of the metrics here. Again, those are retrospective results for 11 clinics doing their things as usual, adding ERA test on top of what they were doing. And we also learn many things from that. For instance, what is the impact of an increase of body mass index? We published this. This was done with our colleagues at Stanford and Baylor. And what we learned here is that there is an increase of the displacement of the window of implantation as BMI increases. And we are about now to publish the second paper about that, by which we found the link between endometrial receptivity and metabolism. Some of the molecules involved in the development of the window of implantation are related to metabolism and metabolic syndrome. So it makes sense that as BMI increases and morbidity, obesity, it has more than 40%, almost 50% of the displacement of the window of implantation. We also learned that this is endometrial thickness is, as we know, is not pathognomonic of endometrial receptivity. So in the optimal frame of 6 to 12, 77% of the patients will be receptive, but 23 they will not. And if they are less than 6 millimeters, well, almost half of them will be receptive and half of them they will not. So again, this is not about thickness, it's not about the phenotype, it's about functionality, and this is the message. Finally, this is a paper that we have done at Stanford with our colleagues, with a group of Steve Quake, by which we have analyzed, we have working through the transcriptomics of the human endometrium, but now going at the single cell level, not just taking the whole tissue, by disaggregating the tissue and having cell by cell analyzing more than 3,000 cells coming from the epithelium, stroma, endothelium, and immune cells. And what we have learned from that is that basically, as we demonstrate in the ERA test, the window of implantation is such an abrupt event that can really be found out even at the single cell level. So you have that at the moment of the window of implantation, the complete transcriptomic signature of many genes completely changes, unlike the decidual reaction in the stromal cells, that is a more progressive function that happens and then stays throughout the luteal phase. But here, the window of implantation is quite drastic, the changes that are produced. So the point that I want to make with endometrial receptivity and what we have to expect in 2018 is just taking you to this concept that when points out the moon, don't look at the finger. And here, the moon is endometrial receptivity is the effect of the progesterone receptor activation in terms of the days of the timing that the progesterone receptor is activated, and then the transcription that is produced in every endometrial cells from the epithelial and the stromal compartment. And this is what is telling us what is the moment that the window of implantation will be there. Don't look at the finger, because the finger is whether we give the progesterone vaginal, intramuscular, or whether the type of progesterone, or whether even the dosage of progesterone. This is pharmacology. It's not going to fix our problems, because this is what we are doing for 20 years. So again, look at the moon and don't look at the fingers. And I would like to just touch briefly, if I can, the concept of the microbes living in the endometrium, living in the endometrial cavity, and what could be the implication of that. I don't know if you are familiar with that, but the Human Microbiome Project is going for about 10 years, and we have learned now that we have 10 times more bacteria than cells. That if you weigh 70 kilograms, 1 kilogram of your body weight is about the microbes that they are living on us, and they are helping very much in the skin and in the gut, in the digestive tract. And many of them, about 20 to 60%, they cannot be cultured with the actual system that we have in the routine microbiological labs. Also, again, this was started a long time ago. We know that 18% of those microbes, they are in the urogenital tract. And we know how relevant is that in the vagina. But we were not aware of what was the relevance in the human endometrium. So we published this last year, by which the aim of that was basically to test the existence of an endometrial microbiota and to know whether if this is hormonally regulated and finally to know what is the reproductive impact of to have an abnormal microbiota in our patients. So just to make a summary of the paper that, of course, you can have access to that, we found that there were some microbes that they were not present in the vagina, and they were present in the endometrial fluid, and the other way around. This was done in the first time in the same patient, vaginal aspiration and endometrial aspiration. Then in about 20% of patients, we have different results. There was no hormonal regulation when in the pre-receptive versus the receptive in terms of the microbiota. And when we tried to study the microbes there, the most important ones, they were lactobacilli, they were streptococci, bifidobacteria, and Gardnerella. And by the percentage of the presence of these microbes, we define what is a normal microbiota, which is a more than 90% lactobacilli and no pathogens, and an abnormal microbiota we call non-lactobacilli dominant microbiota, which is the bad one. And by doing this, these were the results that we published with 30 patients that we did the ERA test on them before doing at the same time that we did the microbiological testing. We followed them, and we learned that in half of the patients, we have an abnormal microbiota. And in those patients with abnormal microbiota, the clinical results were drastically down, even if we were in the same set of patients with the endometrial factor in terms of window of implantation prepared. But the fact that we have adverse microbes there makes that the pregnancy rate went from 70 to 33, that the implantation went from 60 to 23, that the miscarriage was from 16 to 60, and the live birth was, again, this is a short group, it's 30 patients. But in those patients, I think that it was very interesting to see that. And because of that, well, you can see with your eyes what happens in the live birth. The blue are lactobacilli. So this is the profile of the endometrial microbiota when the patient delivers a live birth, when the patient has a miscarriage, or when the patient did not become pregnant. So this is just for a physical demonstration. Since then, we are conducting this validation in a prospective study with different centers worldwide, with about 434 patients with a recurrent implantation failure, trying to see what could be the real impact of this in order to have a test analyzing the endometrial microbiome and being able to counsel about that. In the meantime, there was this publication from a Chinese group by which what they did is to look for the endometrial microbiota, not only in the vagina, as it is known, not only in the endometrium, as we have done, but throughout the complete reproductive tract, the cervix, the tubes, and even the peritoneum. And what they show is that, indeed, the uterine, the cavity, is not a sterile site, neither any part of the female reproductive tract. And there is a microbiota profile which is progressive, is like a continuum along the female reproductive tract. So they are living on us, and they must help, they must be there to help us to produce something or to affect and impede some functions. So in conclusion from this is that the percentage of lactobacilli is important to predict reproductive success. The endometrial microbiota profile, we just divided by LD and NLD. And NLD microbiota, especially the presence of Gardnerella, strongly correlates with the reproductive outcome. So finally, I would like to show you this, because this is a paper that is going to be published now, June 1st. And we have done this work with a group in Italy, Dr. Ettore Ciccinelli from Bari. As you know, he works on endometritis, and so this is the paper here. I don't know if it's over here, and in fact, this is the cover page, which is a very small one in American Journal, is from our paper. So what he did, well, what we did was to go to samples that they have been diagnosed of patients with chronic endometritis and try to see in a blind manner how microbiological testing, what can we add or improve on the actual clinical practice of chronic endometritis. So to do that, we simply obtain the endometrial sample. In this case, we obtain paraffin section from Dr. Ciccinelli, blinded, and we just get the DNA there, and we did either by metagenome, by next generation sequencing, which is what we do the previous analysis, because this is done using NGS, analyzing the 16S ribosomal RNA from the DNA of the bacteria, and then we can get the taxonomy of the bacteria, or just simple RT-PCR for the 9 most important pathogens for chronic endometritis. This is what we try, what we aim to develop. So Dr. Ciccinelli sent us these 113 samples, and some of them, they couldn't be used for different reasons, but at the end of the day, we end up with samples, endometrial samples from 65 patients that he did the 3 routine techniques from chronic endometritis. He did hysteroscopy, he did histology, and he did microbiological culture. And with all of these samples, we did also the microbiological testing. What was very interesting is to learn that altogether, the 3 of them, only they have results in 13 of them, 20% of them. So in some cases, they have agree histology, hysteroscopy, microbial culture, and our molecular biology. Some of cases, they were also positive for that. But in the majority of cases, there was discrepancies between histology, hysteroscopy, between that. This was 2 figures of the paper. So in essence, what I think is important is that only 20% of the patients that Dr. Ciccinelli studied clinically, they agree on the 3 techniques used, okay? So we just took this, and we find out a concordance of 76% of the diagnostic when we apply molecular microbiology. In other words, well, you have here the table. In other words, I think that to go for this microbiological testing to assess for that will be superior than going to one or the other techniques, or you have to have the 3 of them together. So in conclusion from this paper that you can read just in 1 week in the American Journal, and is online, of course, is that molecular microbiology detects and quantifies bacterial DNA from chronic endometritis causing pathogens. The microbiome results using NGS were concordant with RT-PCR in 91% of the cases and coincide with the microbial culture in 75%, allowing for the detection of culturable and non-culturable bacteria. So this molecular diagnosis is equivalent to the use of histology, hysteroscopy, and microbial culture together, overcoming the bias of having any of them in separation. So what I try to show you is, a 360 degrees of what could be the endometrial factor, that the mother is important, the endometrium is there, we have to have a receptive endometrium, but also we may have to look for the absence of contamination, of microbial contamination, of bad pathogens. And I think that learning more about the endometrial factor, we will be better off improving our clinical results. And finally, I want to show the faces of the team working on the ERA, and I have to acknowledge Maria Ruiz, that is the manager of this team, and also the uterine microbiome team, being Inmaculada Moreno, the leader of this team, and my colleagues worldwide working in genomics and trying to bring all of this research to the clinical translation. Thank you very much for your attention.